GenomeQuest DX is healthcare's first clinical decision support system for whole genome, exome, and gene panel diagnostics.
Here we go, it's coming back on, and he's back on again. See how he turns?
Hi, Jonathan. Chocolate sucking.
Yeah.
What you just saw was an example of how early genetic diagnosis of a complex multigenic disorder like non-syndromic hearing loss can enable patients to get an appropriate course of treatment early, in this case, the early insertion of a cochlear implant in a developing brain. GQDX provides such a tool. A patient presents with a set of symptoms or a concern based on family history. Based on the presentation, the medical provider determines that a full or partial genome scan is called for. Patient samples are taken and run through a next-generation sequencing machine in a CLIA lab. This results in next-generation sequencing reads for the patient, which are combined with the world's most up-to-date information on genetic disease and allele frequency data from dbSNP and the 1000 Genomes Project. GQDX analyzes the reads, mapping them to the reference human genome, calling variants, and annotating all of the genomic information with clinically relevant and actionable information. The result is an interactive molecular diagnostic report that is interpreted by a clinical geneticist, a pathologist, or an oncologist. That interpretation is provided back to the provider who uses this information to guide the patient's course of treatment. Let's take a look at GQDX. Here, we are looking at an NGS gene panel of variation related to various forms of hereditary deafness. By exploring this list interactively, we see a particular single base mismatch on chromosome 11 in the MYO7A gene. Unfortunately, this mutation is directly correlated with Usher syndrome 1B, a disease characterized by natal deafness and a progressive retinitis pigmentosa leading to vision loss. 56.4% of the reads aligning to this locus predict this allele. The patient is heterozygous. A review of the literature indicates that this mutation is autosomal recessive. Therefore, the finding is that there is no genetic pathology for the patient, although there is certainly an implication for the family. Any large sequencing diagnostic will generate many variations of unknown significance. These too can be explored interactively. Unfortunately, 343 such variants of unknown significance were found in this patient. Interactive analysis of these variants allows the expert interpreter to consider only those variants which are rare in the general population, have polyphen and SIF scores which predict a deleterious impact on the protein, and which are reasonably well represented in the overall sample. The interpreter presses the Apply button, and the list of variants drops to just one. On further inspection, it falls outside of any gene, and the interpreter has identified no functional implication on the variant. This is GQDX. Analytics are an important element of the GQDX system. Over a series of diagnostic runs, de-identified whole and partial genomes can be used to identify trends in genome variations. GQDX enables the clinical genetics lab to group variations of known and unknown significance by phenotype, ultimately leading to improved diagnostics and a content base which is highly valuable to payers in making determinations on what and how much to pay. GQDX, healthcare's first clinical decision support system for whole genome diagnostics.